Hey, this is Mrs. Reichelt, and this next video covers the cell-mediated immune system response. So the difference between this one and humoral is humoral attacks the pathogens, and cell-mediated is going to actually infect cells, self-cells that are infected with that pathogen. So the antigen must be presented by a macrophage for an immunocompetent T cell. So basically what that means is it's called antigen presentation. So as you have a macrophage, which I'm going to draw a really bad picture here, but let's just pretend that this is my macrophage. And we'll say that this is, I'll do it a little swirly, this is the pathogen. Basically what happens is this macrophage, this good cell, is going to come in here and it's going to engulf that antigen. So ultimately it's going to look like this. This is my macrophage, and it's going to then have that virus or whatever it is, that pathogen inside of it. It's then going to present the antigen. How it does that is it's going to basically take part of that pathogen and it's going to display it on its cell membrane. So you see how normally it's all kind of spiky. I didn't draw the spikes very well over here, but it's kind of spiky. And then you see an area that's kind of spiral. That is called antigen presentation, meaning that it's taking its part of its the pathogen and it's displaying it for all of the other white blood cells to know what it looks like, or all the other B cells. So the T cells will recognize the non-self and self-recognition. So that means that the T cell is going to know the difference between a good cell and a bad cell. Okay, so after antigen binding, clones form as with B cells, but they're different types of classes of cells that are produced. So that means that they're going to make a whole bunch more of these cells, but they're going to be they're going to be named differently from the humoral names from last video. So this is kind of how, I guess this is a better picture than what I drew. So cell-mediated, you basically have this macrophage, and it's going to engulf the antigen, okay? And then it's going to present part of that antigen. So this is the antigen presentation, where it's going to show those helper T cells, hey, this is what the virus looks like, you're going to attack this, okay? Then the T cell is going to be the receptor off of that. We call it a helper T cell because it's going to help. Then from there, you're going to have cytokines that are going to basically mobilize cytotoxic killer T cells. The cytotoxic T cells are going to basically attack infected cells, okay? Basically what happens is once your cell is infected, there's no way that it can be uninfected. There's no repair for that cell. So basically the cytotoxic T cell doesn't really have a choice but to attack that infected cell. In addition, after that antigen presentation that occurs right here, it's going to send information to the B cells so they can now create antibodies that are in that correct shape to hopefully prevent them from replicating inside your good cells. So basically we have two things going on. You have cell-mediated because you're killing your own infected cells. You also have antibody being produced, which is basically humoral, but just be aware that they're not going to happen one and then the other. You're going to have them both, but they're just attacking different things, so they have a different name. So your cell-mediated immune response, you have T cells, which are called cytotoxic killer T cells. They specialize in killing your infected cells. They insert a toxic chemical into that cell, which causes, it's called apoptosis. I'll write that down. Apoptosis, which is programmed cell death. And it does that on purpose. Then you have helper T cells, which recruit other cells to help fight the invaders, and they interact directly with B cells. So T cells and B cells basically work together. Some T cells become cytotoxic, some become helper. B cells, some are just involved in the immune system response, and some of them are memory. So there's some different specific forms of each of these cells, but in general we just call them T and B cells. All right, so here's another kind of diagram to show you kind of what it looks like. So you have a cytotoxic T cell, which is right here, and it's basically going to, we'll say that this is the target cell. This means that this is the one that has the pathogen in it. Okay, so what'll happen is you're going to have all of these cells that are going to, or all of these little pinkish circles, they're going to basically inject the cytotoxic, those toxins, into this target cell, which will cause it to die. Okay, so it has all of these little granules in its different compartments, probably some sort of lysosome or some type of internal compartment. Then it's going to go towards the outer surface and it's going to inject that toxin, which you can see that happening right here, into that target cell. All right, so the T cells are the clones. Then you have regulatory T cells. They are going to release chemicals that suppress the activity of T and B cells. They can also stop the immune response to prevent uncontrolled activity. And then a few members of each clone are memory cells. So you have some T cells that act as memory cells as well. And then the last slide of this chapter, we finally made it to the end here, are the differences between humoral and cell-mediated. So humoral attacks pathogens, cell-mediated attacks infected cells as well as the pathogens. So I hope you found that helpful.